SADC Plant Genetic Resources Center is a network of 16 member states national plant genetic resources centers. It was created to conserve and promote the utilization of indigenous materials in the SADC region.
The SPGRC runs the SADC Regional Gene Bank. The Gene Bank hosts more than 19,400 accessions from the 16 SADC member states. Gene Bank we run is a base collection, meaning that it's storage of seed accessions for the long term. Some of the accessions in the Gene Bank date as far back as early 90s, but they're still viable and have to be viable for a long time until they're used. The materials that we keep in the SADC Gene Bank are coming from the member states. They are duplicated to the Gene Bank for long-term storage. Upon receipt of materials from the member state, the material will be tested for viability to ensure that we are storing something that is viable and not material that cannot germinate. And in that process, there has to be acquisition of this material, which means the material is going to be registered in a Gene Bank management system called the SPGRC Documentation and Information System. We make sure we monitor temperature of the freezer, as we have to store these materials at -18 degrees Celsius. And that means we have to monitor each and every freezer to ensure that materials are stored in an optimal conditions. And to do that, we have a system which monitors each and every freezer and sends a signal to the responsible officer if there was any rise in temperature in any freezer, so that such a freezer could be attended to. That ensures that these materials are optimally stored and can stay viable for a long time.
We package our seeds in two types of packets. We have what we call the distribution pack, or we can also call it a smaller pack. And then we have a bigger pack. So the bigger pack is used for storage. Every 5 years, we'll pick one of the smaller packs just to check what's happening to that seed. In the Gene Bank, we have ventilators and the air cons, which help us to maintain the temperature inside the Gene Bank. And the ventilators, there are two ventilators which help to bring in air, fresh and cool air, and the other one to take out warm and moist air to make sure that the environment is well kept. We have the ex situ and in situ programs. Ex situ program manages the Gene Bank, and the Gene Bank receives materials from member states, that is in seed form, and these are indigenous materials. They are kept outside their natural adaptation in the freezers.
The second section is the in situ conservation. So in situ conservation helps member states with collection of accessions in farms and in the world, and also run the tree gene bank here at SPGRC. The tree gene bank is also called the Arboretum, is an in situ gene bank in which we conserve some medicinal plants that could be of value for use in research. The National Plant Genetic Resources Centers do self-duplication of their materials with the SADC Gene Bank. This is done to ensure that the risk of total loss of germplasm at national level is mitigated. If ever there was a disaster at the member state gene bank and materials were lost, the member state would withdraw materials they have duplicated with SPGRC and continue with their research programs.
Self-duplication is the having of copies of these materials in 2 other gene banks. So we have 2 layers of these gene banks. The SADC gene bank as a first layer or a primary layer. Then a secondary layer is the global seed vault in Norway. So we copy our materials to SADC gene bank, and the same materials are also copied to the secondary layer, which is global seed vault. This is done in order to secure these materials. Whatever happens here, if we lose all the materials, we are able to get the materials either from the SADC gene bank or from global seed vault. The gene bank was established for purposes of conservation and sustainable utilization of plant genetic resources. And also, it is a unit that supports crop development, because the materials that we are conserving in the National Gene Bank are the materials that the breeders can use to breed varieties. From its inception in 1989, as a gene bank, we have managed to collect more than 6,000 seed samples from the farmers across the country. From 1989 to date, we have been keeping these materials, and we plant them, they are able to germinate, meaning that the conditions in which we are keeping these materials are very good conditions, such that the materials are maintained